we're still recapping. Okay, we're okay. But this is what I mentioned at the end of class the other day, is that a mild fever, a lot of people take ibuprofen or other fever reducers when they have a mild fever. That's actually not a good thing. Okay. You monitor a fever. Now, if it gets up above, they usually set it about 101.5. If it starts to get up past 101.5, then yes, it's starting to get out of hand. Okay. But a mild fever, a fever of 99, 99 and a half, 100, 100.5, even 101, it's still not considered to be that big of a deal. But you keep an eye on it. If it starts going up to 102 and 103, then yes, you got to bring that back down, because there is a temperature. It's about 104. Okay, 40 degrees Celsius, 104 degrees Fahrenheit. Once you pass that number, the proteins in your body start to melt. Okay, like the enzymes that perform all the chemical reactions in your body, they have a certain temperature range, and if you go outside that temperature range, they'll stop working. And the high point of that temperature range is about 104. So if you go past 104, then the enzymes in your body will lose their shape. That's called denaturation. When a protein loses its shape, we call that denaturation. And when a protein loses its shape, it ceases to function. And so if you go up past 104, and if you stay up above 104, then the proteins in your body start to lose their shape and they'll stop functioning, and the body will start to break down, and you can go into heat stroke and things like that. So that's the whole idea behind, you want to manage a fever, because it can get out of hand, and so you keep an eye on it. But a mild fever, well, yeah, 104 is up there. That's dangerous. Okay. So yeah, 104, you're like, oh gosh, you need to get that down. So they usually say about 101.5. If you go up above 101.5, you need to start working towards getting it back. Okay. Getting it back towards normal, because you're getting a little bit too far away from normal. You want to go away from normal, but you don't want to go too far. And that happens a lot with positive feedback in general, is that, you're pushing yourself away from normal, but if you push yourself too far, it could actually have a negative effect. And so you want the beneficial effect of pushing your body away from normal, but you don't want to go too far. A lot of parents, a lot of worrywart parents will give their kids ibuprofen or whatever when their temperature's 100. And that's not a good idea, because you're basically suppressing the body's natural built-in response to an infection. A fever is the way we fight infection. And if you kill that, you're taking away one of the body's ways of defending. If you kill a fever, if you destroy a fever with the fever medication. I think I told you guys about that. I saw it. It was a cartoon one time where it had these two bacteria, and on the they were in their little suits, or they were like what they call it, anamorphic bacteria. But they were sitting there talking to each other, and on the wall behind them was a thermometer, and the thermometer read 99.5 or something like that, 100 degrees. And one of the bacteria is talking to the other one. He says, I don't know how much. He's like, what? He's like fanning himself, and he's sweating. He's like, man, it's getting hot in here, Larry. I don't know how long I can last. And then Larry, the other bacteria, goes, just hang on, Johnny, because the mom's getting ready to give ibuprofen. And then it's just a close-up of his eyes, and it says, and we will rise again. Because that's what happens. When you raise your temperature, the bacteria is like, daggone, it's hot. They can't multiply very fast. And then when you kill that, they're like, woohoo, I can go again. So actually giving a fever reducer for a mild fever actually benefits. Think about this. Giving a fever reducer for a mild fever benefits the bacteria. It benefits the infector, because you're going to bring the temperature down, which it likes that. It wants that. It wants the temperature back to 98.6 degrees, because that's where it replicates the best. That's where it infects the best. So you're actually doing the pathogen a favor by giving ibuprofen or other fever reducers for a mild fever. So you keep an eye on it. If it's less than 101.5, just let it go, but still keep an eye on it. If it starts getting up to 102, 103, then yes, you're going to have to bring that back down. That's a little bit too high. That's my advice to you, future parents or current parents. Let your kid fight the infection. It drives us nuts as a parent to watch our kids suffer. And they're sitting there in bed. I feel so bad, Bobby. I just think in my mind they're building their immune system. So they're getting stronger. We hate to see our kids sick, but they have to get sick because of this right here, adaptive immunity. Okay, this is the other form of immunity. Innate is what's built in. Okay, adaptive is the learned immunity. You might hear it called acquired immunity. Okay, it's where you encounter a pathogen of some kind and your body has to figure it out. Your body has to figure how to fight it. Now, once it figures out how to fight it, then it can remember how to fight it later. I think adaptive immunity is really cool because I know your cells don't have brains, but their ability to figure something out, and then once they figure it out, improve upon that and remember, I don't have a mind, they remember, quote unquote, that pathogen for future exposure. And so one of the things you have to kind of keep in your mind about adaptive immunity is to develop it, you have to be exposed to the pathogen. Okay. And that's why a lot of times kids get sick a lot, because it's their first time being exposed to a particular pathogen. Their immune system is developing. Their immune system is figuring it out. And then a lot of times once you develop immunity for that specific, okay, it's more of a specific thing, then you might have immunity to that for years or even decades. But that first time you encounter it, it's a figuring out process. And so one of the differences between innate is just general defense. Adaptive immunity is specific. Okay, you're exposed to a pathogen, and then you have to figure it out, and then now you're on guard against that pathogen for future exposure. Now, what the body's responding to are these sugars and proteins that are on the surface of cells. And so let me pull up a whiteboard here. I got to remember to keep switching back and forth. So let me go to my whiteboard and let's talk about, let's clear that. Ooh. What's going on? Okay, clear all drawings. There we go. All right, so all cells, it doesn't matter if it's cells of your body or if it's foreign cells. All cells have on the surface different sugars and different proteins. Okay, they might be receptors, they may be sugar side chains. All right, so imagine this is a cell of the body. So we'll make him a little happy face. Okay, so this is a cell of the body. So that's supposed to be there. Okay, your immune cells, maybe let's give him a little helmet, a little soldier helmet. All right, your immune cells are always on patrol looking for foreign substances. Our cells display certain sugars and certain proteins. And foreign cells also display proteins, but they might be different proteins. I'm just making drawings up right here. Okay, so imagine this right here is a different cell. Okay, so maybe this is a bacteria or something. So this is the bad guy. Your immune system is supposed to be able to recognize the bad guy, because your immune system can sort of read these sugars and proteins that are on the surface. These are called antigens, okay? Antigens are sugars and proteins that are displayed on the surface of cells, both the body cells and also foreign cells. And so they're called antigens here as well. So antigens are sort of like ID tags that allow the immune system to tell whether a cell is supposed to be there or it's foreign. If it's the antigens that your cells display, those are called self-antigens. And the immune system is not supposed to attack them. All right. The ones that are displayed by foreign cells are called foreign antigens. That is the one that the immune system is supposed to recognize as not supposed to be here. And so what your immune cells do is they kind of bump up against your other cells. And when they bump up against them, they sort of read the surface and check the ID. It's really, I always think of antigens as sort of like the ID that a lot of us have to wear at our job. I mean, some of you guys may work in a hospital setting where you have to wear your ID badge around your neck on a lanyard or something. And whenever you walk up to a particular area, you got to show your ID and say, I'm supposed to be here, right? And if you're not showing that ID, then you're not going to be allowed in to whatever, or you may be thrown out. You may be attacked and thrown out. So a lot of jobs require ID tags. You know, when I work at school, I'm supposed to wear an ID tag that says Bridge Valley Community Technical College, Charles Pomeroy. Okay, and that's my little ID tag that I wear on my shirt. Depending on where you work at, you may have to wear ID tags. Antigens are no different. It's basically what your cells are saying. Hey, I'm supposed to be here. I have the right antigens, while the foreign cells don't display the correct ID tags. So it's almost like your immune cells are bouncers at a club reading IDs. And if they come across one that doesn't jive with them, then they will go, nope, you're not supposed to be here. You need to go. And they will attack and destroy that foreign cell. And so this whole idea of antigens is extremely important. It actually comes into play later on with blood typing, because depending on what blood type you are, your red blood cells display different antigens. You might display the A antigen on your red blood cells, and we call you type A. You may display the B antigen, and we call you type B. You may display both, and we call you type AB. But that comes from what antigens are you displaying? What ID tags are your cells putting out there? But the immune system is supposed to recognize any foreign antigen and attack it. And so what your lymphocytes, and these are the ones that are involved with adaptive immunity. Okay, so when you talk about adaptive immunity, you're talking about a specific type of white blood cell. They're called lymphocytes. And I mentioned them a little bit on Monday, that you have what's called T cells and B cells, or T lymphocytes and B lymphocytes. Those are the ones that are involved with this figuring out process of adaptive immunity. They are looking for a particular antigen. They're looking for the enemy ID. They're trying to spot the fake ID, so to speak. I always laugh at this woman. Does this make sense, by the way? Does anybody need any clarification here? You know, antigens are basically ID tags that cells display. Your immune system can recognize what's supposed to be there and what's not. This will come back into play here in just a little bit. So I'm going to leave this drawing up and go back to the PowerPoint. But this picture of this woman, it makes me laugh. I don't know why they chose this particular picture. But this woman, you know, she's got like all of her organs exposed, and she's looking at you all sexy. She's like staring a hole in your soul. But I always thought that picture was funny. Let's see, T and B. Okay, now remember how the whole idea, we talked about this the other day. All your lymphocytes, T cells and B cells, they're all made in the bone marrow. And then they have to undergo a maturation process. They have to develop the ability to fight infection. We call that immunocompetence. B cells stay in the bone marrow. So they're produced in the bone marrow and they mature in the bone marrow. And so you can look at the B as standing for bone marrow, B for bone marrow. The T cells are produced in the bone marrow, but they have to travel to the thymus, which is located above your heart, because the thymus produces hormones that help T cells mature. And so that's why they're called T cells, is because they undergo their maturation process. They develop their immunocompetence in the thymus. Now, once you have mature B cells and mature T cells, they will leave the primary lymphatic structures, the bone marrow and the thymus. They will leave and then go into your lymphatic tissue, colonize your lymphatic tissue and organs, and set up shop. Now they're ready to go, and they're just waiting for a foreign antigen to show up. So they're going to go and colonize the lymph nodes, the tonsils, the spleen, the other lymphatic tissue. They're going to go and set up shop and wait for the enemy. They have to be exposed in order to figure things out. Just like, a lot of times that first battle in a war where you're figuring each other out, usually doesn't go well for the two sides because you have no idea the capability of your enemy and you have no idea how to fight it. And so that first battle is sort of like a figuring out process. It's no different with adaptive immunity. The first time you're exposed to a pathogen, you're going to get sick usually. And it may take you days or weeks or even months to fully get rid of that infection because you got to train them. You got to train them through battle. But once you train them through battle, they get better and better. Just like you have soldiers, right? You have the rookie soldiers who have never been in a battle before, and then you have the battle-worn soldiers that are in like their 5th tour of whatever. And so that's kind of what's going on here. The first time you encounter, it's a little difficult, but then you get better at it and you get more efficient with it. Same thing's going on with adaptive immunity. T cells kind of run the show. Okay, there's different kinds of T cells, but they're sort of in charge when it comes to adaptive immunity. They have to know, they have to be kind of told, though, that there's an enemy present. So the first cells that kind of come into play are these cells called antigen-presenting cells. And the shorthand is APC. So sometimes when you read a chapter in a book about immune, you might see APC. That stands for antigen-presenting cells. These are cells that basically find the antigen and say, hey, look what I found. It's about the best way you can describe antigen-presenting cells. And an example, so let's go back to our whiteboard here. An example of an antigen-presenting cell is macrophages. Okay, macrophages will do this. So pay attention here. You got your macrophage. You've got your bacteria. So your macrophage, remember that monocytes are the largest white blood cell. And so they're going to be, typically they'll be a lot bigger than a bacterial cell. So here's the bacterial cell here. That's the foreign cell or the foreign substance. A macrophage will swallow it up. So we call that phagocytosis. And what it does is it kind of reaches out its membrane and then brings it in. Okay, so the macrophage is going to bring in that foreign substance. He's going to swallow it. So we call that phagocytosis. So he's going to swallow it up. Oh, wow, how did I do that? He's going to bring it in. I don't like that. It's too thick. There we go. So he's going to bring it in, and he's going to break it down. Okay, so he's going to basically digest it. All right. So he brings it in, and he digests it. He breaks it down into little pieces. He's going to take one of those pieces, or several really, but he's going to take these pieces that he creates after he breaks it down, and he's going to manufacture a protein receptor. And it's called an MHC. Okay, so this is a protein receptor that the cell produces. MHC. He's going to link a piece of that cell, which is basically an antigen. He's going to link a piece of that cell to that receptor. He's going to move the receptor to the surface and display the antigen. Okay, so he brings it in, he breaks it down, he hooks it up to a protein receptor called an MHC. He moves that receptor to the surface and he displays the antigen saying, hey, look what I found. That's basically what they do. Look what I found. And so let's recap that process one more time. All right, so here's your foreign cell. Here's your macrophage. Macrophage is going to bring in the foreign cell. He's going to break it down into pieces. He's going to take a receptor called an MHC. He's going to take those particles, attach them to the receptor, move it to the surface, and display it. And what he's basically saying is, hey, look what I found. So these are kind of like, I guess you can consider antigen presenting cells as sort of being like the radar people. They find the enemy and they're like, come here and take a look at this. I found something. I don't know what it is. I found something. Take a look at this. And then what you have happening is you have T cells, which are basically your scouts. Okay, they're called helper T cells. These travel throughout your body, checking what the antigen presenting cells are saying, look what I found. So one says, look what I found. The other one comes up and checks it and says, yes, that is a foreign substance. We need to do something about this. And so just like you have the radar detector say, Colonel, come here and look at this. I found this on my radar screen. What is this? What do I do? Or, should I proceed? That sort of thing. And then the superior officer comes up and goes, yes, that looks like an enemy sub or whatever. I need to go get help. I need to let everybody else know. That's what helper T cells do. Helper T cells activate everybody else. So they come up and kind of do the confirmation process. Sometimes I always think sometimes the antigen presenting cells are like, look what I found. And the T cells like, that's nothing. I'm not, that's just a foreign particle that you found. That's no big deal. But if they do recognize it as being something dangerous, then they will activate the other immune cells. So macrophages display antigens, saying, look what I found, on these protein receptors called MHCs. Then you also have these cells in your skin called dendritic cells. And they do the same thing. They'll come across the foreign bacteria, they'll swallow it up, they'll break it down, link it to an MHC, display it on the surface, and say, look what I found. That's all they do. They don't decide on a response. They don't activate anybody else. Their job is to just say, hey, I found this. Is it anything important? It's a T cell's job to see if it's important or not. Does that make sense? Anybody have any questions about that? The reason they're called MHCs, the full name is major histocompatibility complexes. All right. Histocompatibility means tissue compatibility. These MHCs are very important when you're trying to match an organ donor with an organ recipient. All right. Because your immune system sort of interacts with these MHCs. And if the recipient's immune system reacts negatively to the MHCs on the donor's organ, then it will reject the tissue. And so that the match tissues between donor and recipient, the MHCs really come into play with that. Because if you don't have the same MHCs as the other person, then you don't match. And so that's the whole idea. That's why they call them histocompatibility complexes. They use it to measure tissue compatibility between donor and recipient. Let's go back. Whoops. There we go. All right, somebody just tried to get in, but okay. There we go. All right. So the antigen presenting cell will swallow up the antigen, link it to an MHC, display it on the surface, and basically say, look what I found. The job of checking that are these cells called helper T cells. Helper T cells are the scouts of the immune system. They go around checking antigens that are being displayed, and if they think that it needs to be dealt with, they will activate other immune cells. And so these, especially helper T cells, are very important for immunity. One, they find the enemy, they recognize the enemy, and they activate other immune cells. A lot of other immune cells don't do anything until they're activated by a helper T cell. So the helper T cells sort of initiate and oversee immune responses. They're extremely important. And so I put that helper T cells patrol around the body looking for the enemy. They go up to antigen presenting cells and they check what they're showing them. If a pathogen is found, helper T cells will initiate an immune response. And then C is very important. They activate other immune cells. Like I said, some immune cells don't do anything unless they're activated by a helper T. Now, the helper T cells, the scouts of your immune system, those are the ones that are destroyed by HIV virus. HIV virus attacks your helper T cells and kills them. If you don't have your scouts, it's very hard for you to mount a good immune response. If you don't know there's an enemy out there, then you can't do anything about it. If you can't activate the other cells, then you're not going to get a good immune response. And that's the job of the helper T cells. And that's why AIDS is so devastating. It basically wipes out your immune system by destroying these cells. These cells run the show. They initiate immune responses and they oversee immune responses and activate other cells. So if they're not working, your immune system is severely compromised. Okay. People that have AIDS don't die from AIDS. They die from infections. And a lot of times these infections or cancer, but a lot of times these infections or cancers, if the person's immune system was healthy, they'd be able to fight it off with not really any problem. So a lot of times people with AIDS die from stuff like pneumonia. I said ammonia, pneumonia. But you know, some of these infections that are mild, yeah, we might get sick and things, but we can usually fight it off within a week or so, right? But when you have AIDS, you can't fight off infection, and so you die from infections of different kinds. It severely weakens your immune system because it kills your helper T cells. So that's their job, run the show. Now, you also have these cells called regulatory T cells. Their job is to stop an immune response when the infection's over. You know, you want your body to produce an immune response when there is a pathogen present, but you want to be able to stop the immune response when the infection is under control. And that's what regulatory T cells do. They suppress the activity of other cells. They do it to end an immune response, but they may also be involved with making sure that your immune system doesn't go overboard. Okay. For example, an allergic reaction. A lot of times an allergic reaction is your immune system sort of freaking out, going way overboard with its reaction. And so they think that regulatory T cells also help to sort of make sure that you don't have allergic reactions happen all the time. And then you have the coup de grace, I think, of immune disorders is autoimmune disease. I think autoimmune disease fascinates me because it's your own body turning on itself. Your body's immune cells will attack your own cells and kill them. And that's what autoimmune disease is. You're imagine that. Imagine like waking up tomorrow and the army has invaded Washington, D.C. and destroyed the government. The US Army has. You're like, what? They turn rogue on us. They turncoat. That's what autoimmune disease is kind of like. It's like your body's own defense network turns on itself. Allergies is similar. It's just your body freaks out, and what was supposed to protect you actually does more harm than good. Both of those kind of fascinate me. If you can't trust your immune system to defend you, then you're in trouble. And if it turns around and attacks your own cells, that's just the worst. But that's what regulatory T cells do. They kind of make sure that immune responses don't get out of hand or don't keep going longer than they should. Let's see, killer T. I always like that name, killer T. They're also called cytotoxic T cells. But what these cells do, I like these guys too. These are the mob hitmen of the body. That's why I always describe them. One of their primary job, killer T cells, is to kill your own cells when they need to be killed. All right. They notice what it says here. They have to be activated by helper Ts. They don't do anything unless helper Ts tell them to. But they kill other cells of the body that need to be killed. Cells that have become infected and cells that have become cancerous. So they actually kill your cells, your own body cells, when they have to be killed. It's kind of like that old had to be done. Johnny had to be shut up. He was talking too much. You have those people in the mob. Their job is to kill members of the mob because they're talking too much and they need to go. And that's kind of what these guys do. I always imagine an infected cell coming up to these and going, help me, I'm infected. And he's like, yeah, you're infected. You got to go. And he just kills them. And then he's like, had to be done. You know what I mean? It's like one of those type deals. And so, these are the ones that stab your cells in the back. It's like, oh, it's okay. And he kills them. It's like, sorry, you was infected. I've got to kill you before you infect everybody else. You see those zombie movies where once somebody gets bit by a zombie, they need to be killed because they're going to turn into a zombie too. So you got to kill them. It's kind of what killer T cells do. They come across an infected cell, they're going to kill that cell so it doesn't infect everybody else. Same deal. They're like, you got to go. If a cell becomes cancerous, you got to kill it before it forms a tumor. Infected cells and cancerous cells will display new antigens, that your body sees as being, that ain't right. You know what I mean? So imagine that when a cell becomes infected, it starts displaying different ID tags. It's basically saying, hey, I'm infected. And then the body's like, you got to go before you infect everybody else. Or a cancerous cell has to go because it'll form a tumor. So got to kill it. Now, the way they kill it is by stabbing it. So it literally is like, they give you a hug and then, I knew it was you, Frodo. And then, or Fredo. I said Frodo, that's Hobbit. I knew it was you, Fredo. And then stab him in the back. That's Godfather. So I got my Godfather and Hobbit mixed up. Frodo. Yeah. But basically, you kind of go, it's okay. And then you just slice their neck and say, you got to go. But that's how they do it. They actually release these substances they're called perforins. Okay, perforins are these chemicals that are released by these killer T cells. And what perforins do, okay, the word is perforate, which means poke holes, right? So when you have perforated paper, it's paper with holes in it. And so what these perforins do is they poke holes in the membrane of a cell. And when you poke holes in the membrane, the cell will basically fall apart and die. And so these are the ones that kill with hand-to-hand combat. You got to get close to the other cell. Keep your enemies close. Keep your friends close, your enemies closer. But they get close to the cell. They release those substances called perforins. It'll poke holes in the cell and cause it to die. And they walk away, clean off their knife, and say it had to be done. I wish these videos would work. I'm gonna try to find videos this weekend. I forgot that Flash Player doesn't work anymore. You know what I'm talking about? Adobe Flash Player. These little hyperlinks here, these blue hyperlinks on these slides, they went to videos, but they were Adobe Flash Player videos, and they don't work anymore. I just realized that like yesterday, so I was like, I'll just try to find something this weekend that could maybe show it a little bit better. Then I can share it with you because it's good to kind of see these guys in action. And so these guys stab with bayonets. They poke holes in the enemy. It's not really the enemy, it's your own cells that got to go. And so you got helper T cells. They are going to go on patrol, find the enemy, let everybody else know the enemy's there, initiate the immune response, oversee the immune response. Regulatory T cells are designed to keep an immune response from getting out of control. And then your killer T cells are the ones who kill your own cells when they have to be killed. They're talking too much, they're infected, they're cancerous, they got to go before they cause major problems. You know, don't underestimate, and I think I mentioned this to you guys the other day, don't underestimate one of the important things your immune cells do, your killer T cells, is they kill your cells when they become cancerous. Okay, they're very important for making sure that cancer doesn't occur in your body. I think I mentioned this to you guys the other day, I may not have, but every day there's a chance for cancer to form in your body. Because all cancer is mitosis gone out of control. So whenever one cell starts to divide, okay, it's supposed to divide to replace lost cells, and then it's supposed to stop. But if it keeps going and it starts, dividing out of control, that's cancer. And so anytime mitosis occurs, there is a slight chance that cancer could occur. And so every day, theoretically, there's a chance that a cancerous cell will form in your body. Your immune system is supposed to recognize it and kill it before it forms a tumor. Now, if your immune system's weakened, that's a lot of times when cancer shows up. If your immune system was healthy, you could have recognized it and killed it, no big deal, it's over. But if you have a weak immune system, then sometimes those opportunistic cancers will show up. And so you have cancers developing at a higher rate in older people because their immune system is weaker. There's actually a form of skin cancer called Kaposi sarcoma, which is seen a lot in people with AIDS. It's a opportunistic skin cancer that develops in people with a weakened immune system. If you take immunosuppressants, then your immune system is going to be weaker and the chance for cancer increases. And then stress. Remember we talked about stress? Stress produces cortisol. Cortisol weakens your immune system. And maybe one of those cells that would have been caught by your immune system, one of those cancerous cells that would have been caught and destroyed, makes it through the checkpoints and you develop cancer. So anything that weakens your immune system makes you more prone to infections and cancer because it's going to suppress these killer T cells. That's their job. Find cells that have gone rogue or find cells that have to be removed or else you're going to have bigger problems. You're going to have a worse infection or cancer. Anybody have any questions about T cells? The other cells are called B cells. Now they attack from a distance. They can help kill a foreign cell without being anywhere near it, okay? Because what they do is they produce proteins that they then release, and those proteins are what travel through the body. And so they may produce, they're called antibodies. But antibodies are basically proteins produced by B cells that circulate, and they will attach to the foreign cell. They'll attach to the antigens. They're designed to match antigens. And so these proteins will bind to a specific antigen, and it will sort of trigger an immune response. I always look at antibodies as kind of like kill tags. If an antibody sticks to a cell, that basically activates your immune system to kill it. You're basically tagging it saying this needs to go, okay? So B cells are kind of like the bombers or the snipers of the immune system. They attack from a distance. They produce their antibodies. Now the fancy term, and you guys that are going into medical lab technology, you'll probably hear it called this more often. Another name for antibodies is immunoglobulins. So you have like immunoglobulin A, immunoglobulin E. Those are just different types of antibodies that you'll hear about, and that's what they're called on blood tests, usually, anyways. A lot of times on blood tests, there'll be immunoglobulin something. And so it's just another word for antibodies. These are proteins that float around in the blood, and they're designed to stick to foreign cells and sort of tag them. And when they get tagged, that means, hey, this isn't supposed to be here. Take care of it. And so it says antibodies help fight infection in a few ways. One is antibodies will activate what we call complement proteins, which are proteins that are also involved with an immune response. And to sort of keep it just more general, I just put that complement proteins as a whole, complement proteins enhance immune responses. They make immune responses stronger. So they strengthen immune responses. We don't have to get into what each type of complement protein does and all that good stuff. We can just kind of sum it up in they strengthen immune responses. So one of the things that antibodies do is activate those complement proteins. They also cause cells to stick together, and that's called agglutination. So agglutination, and this is actually, you can kind of see it here down the bottom. Imagine you have this immunoglobulin G down here, which is shaped like this right here. Each one of these here, here, and here can stick to a cell, a foreign cell. So imagine you have a cell here, it gets stuck there, so it's been tagged, right? It's been tagged to be killed. And then you'll have a cell stick here, then you'll have a cell stick here. And then you might have another antibody grab a hold of him, and then it will also grab a hold of this guy and this guy. And then you might have an antibody which sticks to like him, him, and him. And then what you have happening is over time, all these cells will kind of start sticking together as they bind to the different antibodies, and it kind of makes a little glob of cells. And they call that agglutination. It's when foreign cells stick together. Now, what that does is a few different things. One, it keeps these cells from spreading out and infecting. If you can contain them, right? It kind of contains them together so they're not spreading everywhere and causing infections all over the place. So it sort of helps limit the spread by holding them together in a confined space. But also it's a lot easier for your immune cells to find a large enemy versus a small enemy. So as this raft of cells floats down your blood or whatever, your immune cells are able to attack it more easy. So it just makes it more easy for agglutination, makes it more easy to limit the spread, but also for your immune cells to find it and attack it. Agglutination actually comes into play in lab today, because that's what happens when you put the wrong blood into somebody. All those red blood cells stick together and cause like a massive body clot. But it's agglutination. It's them sticking together because they're binding to antibodies. And then they're kill tags. They say that macrophages, the cells that eat other cells, macrophages like their cells buttered with antibodies. Whenever a cell has a bunch of antibodies, so imagine you have a cell and it's got all these antibodies stuck to it, that basically is going to trigger the macrophage to eat it. It's like adding salt or something to it. The macrophage is like, yum, yum. He likes to eat his buttered with antibodies. And so that's like the three. Now this could be, and like I told you, I'm going to make a list this weekend of possible like short answer questions. This one will be a good one to ask you. Like list 2 ways or list 3 ways that antibodies help fight infection. 1, they activate complement proteins, which enhances the immune response. 2, they cause agglutination of cells, which means the cells don't spread as easily. And then 3, they tag the cell basically with the kill tag. saying this needs to be swallowed up, this needs to be destroyed. It'll be on that list I make this weekend. Be able to list at least 2 or 3 ways that antibodies help fight infection. And they're listed there in your notes down there on number 5 underneath B cells. We still got about 5 minutes, so we can get through. We can get through the rest. We'll save the whole active, passive, and allergies. We can save all that for next Monday. But let's finish up with B cells. When B cells are activated, they will differentiate into 2 types of B cells. So just like you had T cells, and then you had helper T, regulatory T, killer T. Those are all just different types of T cells. Well, with B cells, there's 2 different types of B cells. Whenever you have an infection going on, you're going to get the production of both of these types of cells. And it's basically what is their job? Okay, plasma cells, they're a type of B cell. Their job is to fight the current infection. So they will differentiate into plasma cells, and they will start producing antibodies that will attack the cells now, the foreign cells. But then you also get the production of these guys called memory cells. Memory cells don't deal with the current infection. They actually stay dormant. They don't fight now. But they're created, and then the next time the pathogen comes in your body, they are ready to go. And so you produce cells that will fight now, and you produce cells that will fight later. The plasma cells fight now, the memory cells fight later. Now, the production of the memory cells is why you get prolonged immunity after you experience that pathogen for the first time. Okay? So, yes, a lot of times when you first encounter a pathogen, you have trouble figuring it out. You may get sick. You may be sick for days, weeks, or even months. But what you're doing there is, as you're fighting that first time, you're developing those memory cells. The next time that pathogen comes in your body, you're able to mount a much quicker and a much stronger immune response. That's the whole idea behind developing the immune system. That's the whole idea behind adaptive immunity, is the creation of these memory cells. So that way they're ready to go for the next time. That's why kids get sick a lot. And that's why you need to let kids get sick, okay? Because as they're fighting that infection, they're going to be producing memory cells for the next time. So kids got to get sick. I mean, the Native Americans used to take babies and take them around to the sick people in the tribe and introduce them to certain, illnesses. So if you had somebody in the tribe who had a cold or something, you would actually have them hold the baby to go ahead and get the baby sick. It was basically a natural inoculation type deal. Go ahead and get them exposed. Go ahead and get them developing those memory cells so they'll have immunity later. That's why when your kid has your kid's friend has chicken pox, you're like, go play with them. They used to tell us all the time when he was a kid, Johnny down the street has chicken pox. Go play with him. Go ahead and get chicken pox. You're going to get it anyway, so go ahead and get it now and get it out the way. Because whenever you get chicken pox, you get it, right? But you produce those memory cells, and so most people get chicken pox once. It might flare up later as shingles, but same virus. But usually you get chickenpox once and you're good, because once you've developed those immune cells, you get decades of protection. The production of memory cells is the whole point behind vaccination. You introduce the pathogen in a modified form, because you want the body to react to it and produce memory cells. So that way, when it really comes in your body later, you've already got your memory cells ready to go. And we'll kind of come back to this on Monday. We'll come back to this whole idea that, this whole training of your immune system. One of the most important things is you got to be exposed, whether it be through natural exposure or whether it be through vaccination or inoculation procedures. But you have to be exposed to something to get the creation of memory cells. And then once you have your memory cells, you might have years of immunity, decades of immunity, or even in some cases, lifelong immunity to a particular virus or a particular bacteria. Sometimes you have to, that's kind of hard, it's kind of weird to say this, sometimes you have to remind your memory cells that something's there, and that's why we get boosters and stuff, is you kind of, every once in a while, you kind of introduce it again and say, hey, this is still out there. And that way your memory cells stay trained to recognize it when it comes in the body for real. So the production of memory cells is extremely important when it comes to adaptive immunity. You don't have those memory cells the first time you're exposed. That's why it's difficult. But once you figure it out, then you can have immunity for a long time after that, depending on the virus. The reason why we have to get flu shots every year is because the virus mutates so quickly that one flu strain one year is not the same flu strain next year. But some don't mutate fast, and you may get a particular cold strain or a particular virus one time, and that's it. Fight it off, create the memory cells, and you're good to go for years, decades, or even the rest of your life. We'll kind of recap this because this goes into the next section, but we'll do that on Monday. So Monday we'll pick up here. We'll finish up immunity, and then we'll go right into respiratory system. The respiratory system notes are already in Blackboard. If you go to the folder that says Chapter 23 Respiratory, the typed up notes are in there. And so sometime this weekend, print off those notes and bring them with you Monday. We'll finish immunity and then go right into to respiratory. In between now and 1 o'clock, grab that bag with the blood typing supplies. And what we're going to do is when we get back from break, we will kind of go over the concepts of blood typing. The actual doing the blood typing procedure takes like 5-10 minutes, but you got to understand sort of like the background information of why certain blood types can't give to other blood types and things like that. So we'll do all that when we get back from break. If you need to talk to me, stick around. Otherwise, I'll see you back about 1.
So I have a comment to share.
Okay, is it private?
No. So I started reading the Cells at Work manga.